Hello, can everyone hear me?
Yes.
Okay, thank you. So, welcome to the second session of the showcase in Track 1, and this is going to be a session on health sciences. Can you all see my screen, or can you not see my screen?
You're not sharing your screen at this time.
You are not. I am not. Okay. All right. So let me see if I can, let's see. Get back
It
here.
was a second ago, if that helps.
Okay. Let me see if I can. So are you seeing a bunch of screen, like a bunch of Zoom windows, or are you just seeing this, the showcase?
We just see your profile photo at this moment. But earlier we were able to see the slide.
How about that?
There we go.
Okay, yay. Okay, so welcome. So just a few housekeeping announcements. So just as a reminder, this presentation is being recorded. Student panelists, just a reminder that you do have 10 minutes, and that is total. That is with your presentation and Q&A. We do encourage questions from other panelists and our audience, and you can ask those questions via the Q&A button located on your toolbar. We have disabled the raise your hand, so please don't try to use that, okay? All right, so without further ado, let's get started. And Cameron will be presenting first. Cameron, are you ready to present and share your screen?
Yes, ma'am.
Okay.
Just to make sure, can you see?
Yes, I can see it.
Okay, perfect. So hi, everybody. My name is Cameron Scheitzach, and I am a junior here at UK, and I work under Dr. Fry in the Center for Muscle Biology in the College of Health Sciences. And today I will be presenting a poster on skeletal muscle dysfunction and recovery following an ACL injury. ACL injuries are the most common injury among athletes and young people. It involves the atrophy of the quadricep, which is an important muscle for just everyday lifestyle, for extending your knee, for straightening your leg. And so with this, quadricep atrophy is a significant contributor to weakness and is not restored completely with surgery or rehabilitation. Furthermore, with the quadricep weakness is associated with diminished quality of life, increased injury risk, and premature development of osteoarthritis. And so when it comes to understanding muscle fiber and just the whole thing of ACL injury, you have to understand two key terms, which would be hypertrophy and atrophy, which is located in figure 1 right here. So in the middle, you see a just a regular muscle fiber size. And then to the right of that is an example of hypertrophy, which is defined as those muscle fibers that add myonuclei to support an increase in the fiber volume. In contrast of that, you have atrophy, which is associated with ACL injuries that reduces the size of that muscle fiber. And so this figure really illuminates the overarching theme of the unknown territory of this subject, which is whether the loss of muscle size is accompanied by a loss of myonuclear content. And so that again is illustrated in this figure right here. And as you can see, these question marks are relatively close to these myonuclei, illustrating what is happening to those myonuclei and what is happening to the myonuclear content. So this figure had launched the purpose of this study, which was to analyze isolated fibers from skeletal muscle biopsies collected from patients following an ACL injury. My goal for this project was to determine muscle fiber volume, myonuclear density, and also the myonuclear domain of a muscle fiber. And so further from this, hopefully identify further development of rehabilitation techniques. And so how I went about this study is I received 10 biopsies, or I received biopsies from 10 human study participants. 6 were female, 4 were male, and the average age was 19 years old. And so I received biopsies from both the injured and the non-injured leg. And the non-injured was the genetic control within this experiment. So after receiving these biopsies, I mechanically and chemically digested these samples and then further stained them in DAPI, which is, as you can see in figure 2, DAPI is the staining in blue here that highlights the fiber itself underneath the microscope and allows me to easily point out the myonuclei number within that fiber. And so I would take these samples under the microscope. I would image 10 for each the injured and non-injured leg. And then following this, I would calculate the fiber volume, the myonuclear density, and the myonuclear domain in a data sheet. And then this data was extracted and analyzed and paired with a t-test in which the statistical significance was defined as the p-value less than 0.05. So just to reiterate, the muscle size was measured in a single fiber volume of cubic micrometers. The myonuclear domain was measured by the volume per myonucleus, so cubic micrometers per myonucleus, and the myonuclear number was number of myonuclei per 100 cubic micrometers. And again, these were analyzed for 10 fibers per limb of both the ACL reconstructed and also the healthy limb. And again, this is just an image of what you would see if you would look under the microscope, which I think is a pretty cool image of a single muscle fiber, again, stained with DAPI. And so directly below figure 2 is figure 3 that highlights 2 important graphs, one illustrating the volume and one illustrating the myonuclei number. And so in the volume graph, you can obviously that the ACL reconstructed limb was significantly lower than the volume of what we saw in the healthy limb. More so, the number of myonuclei per 100 micrometers was a little bit higher in the ACL reconstructed, averaging about 22 per 100 micrometers than in the healthy limb, where they were averaging about 21 myonuclei per 100 micrometers. And so from this data, I was able to calculate a further graph in figure 4 that illustrates the myonuclear domain. And so this compares the myonuclear domain found in healthy and the ACL reconstructed, in which I found that the ACL injury is associated with a significantly lower myonuclear domain. As shown, as I saw in figure 3, there's a significant loss of muscle fiber volume following an ACL injury that is not associated with the myonuclear number. Furthermore, this illustrates a relationship between the myonuclear domain that is significantly lower in the ACL injured limb. And so from this data, I was able to conclude that the fiber volume was significantly lower in the ACL reconstructed limb, but this atrophy was not associated with a loss of myonuclear density. And so this really illustrates the recent research that shows there's an ability to preserve this myonuclear density during muscle loss, which may enhance muscle growth. Furthermore, this allows us to tap into the idea of further development of regeneration rehabilitation techniques. Our prior work showed deficits in muscle stem cells with their ability to add myonuclei to the fiber. So with this and understanding the sustainability and maintenance of myonuclear domain and density, that could lead to and contribute to future rehabilitation techniques for individuals who suffer from ACL injuries. And that is it.
Nice job, Cameron.
Thank you.
I think at this point, anybody who has questions, please feel free to ask Cameron. Jean, did you have a question for Cameron? Cameron, there is a question in the queue for you.
Oh, sorry.
That's okay. Can you see it, or would you like me to read it to you?
It just says cannot see.
Okay. The question is, what was your favorite part of working in the lab and doing this project?
I think my favorite part was just being able to see the results and the images on the microscope itself and seeing how everything just accumulated and how important this is and how necessary it is. I really never knew the research field before, and stepping into this, and Dr. Fry and his colleagues have done a great job of bringing me in and teaching me. And so just being a witness of this and being a small piece of a large project that could really help people in the long run is just my favorite thing.
And there were just a few more comments. I don't know, Cameron, if you can see them, but Dr. Andretti said wonderful poster. And then Camille said really great work and presentation, Cameron.
Thank you. Appreciate it. Thank you so much.
Are there any more questions for Cameron and before we move to the next presenter? Okay, well, thank you and great job, Cameron.
Thank you so much. I appreciate it.
Okay, our next presenter is Amy Keith, and Amy works under the direction of Dr. Rhonda Reamer. Amy, are you ready to present?
I am, thank you.
Awesome.
Okay, hello everyone. My name is Amy Keith, and like Ms. Russell stated, I will be presenting my research project, which was under the direction of Dr. Reamer in the College of Health Sciences. And today I'll be presenting to you distinctive and unique people, healthcare students' discomfort in defining diversity. Okay, so a little bit about what's been done in the past is that there have been a lot of qualitative research projects on diversity courses, but not necessarily about healthcare and not necessarily longitudinal research. So some of these include white women in a semester-long diversity course, also service learning diversity course for pre-education majors, but like I said, not necessarily in the healthcare fields and not over a designated time period, so not longitudinal. So our research aimed to fill these gaps by providing longitudinal research over the course of a semester, and we had 3 time points of data, as well as a diversity course in pre-health students. Okay, so the questions that guided our research were: how do healthcare students understand, connect to, and define diversity? What are the effects of such a course on views of cultural competency and diversity in pre-healthcare students? And what are their thoughts, behaviors, and feelings surrounding diversity and cultural competency throughout this course? So our methods included the cultural competency course itself. So this semester it consisted of 30 undergraduate students. Most of these were white, female, upper middle class students. It had a service learning component as well as small group discussions that were not for a grade. It was made very clear to the students that this portion of the course was to kind of debrief from the lecture, to go through those thought processes and to not to have this part of the course for a grade. We also had experiential activities. So activities that would expose this class to circumstances that they might not have been exposed to before, such as food deserts, taking public transportation to grocery stores, that sort of thing. And then there were 3 assignments. So this was the longitudinal aspect of this research. So the first time point of data was before the start of the course to kind of see where students were coming from and what their preliminary beliefs were about cultural competency and diversity. At the midterm, this was to kind of see what the first part of this class had an impact on those thoughts, and then finally to look at those other time points of data to see how far they've come, and then to also decide where they wanted to go from there. And so we use thematic analysis to kind of go through these time points of data. We use transcription, line by line initial coding. So going through highlighting different phrases within the written data and using gerunds to describe the thought processes and connect to broader theories. And then we took that data and we even further focus coded it. So we kind of disseminated that down to a few less codes, and then searched for themes and sub-themes within those, and then refined and named these themes. So I will be presenting to you about the themes and sub-themes that we uncovered. But before I do this, I do want to go ahead and address my subjectivities. It's very important to disclose this, especially in qualitative research, because this could have had an impact on the themes that we uncovered. And so I am a white heterosexual cis woman. I am an undergraduate student who studies social determinants of health and cultural competency, and therefore I place those at and value those very highly in my personal and professional lives. I will have a future in public health. I'm pursuing graduate work specifically on this subject. And then I grew up in a rural area where cultural competency and humility were not valued in healthcare specifically, which is why I wanted to do this research. Okay, so back to the research. This is kind of just like an overview of the themes that we saw. So time 1, this was before the class meeting. That was characterized by experiencing discomfort. And then time 2 was the midterm of the course, and that was characterized by reacting to their initial discomfort. Time 3 was the final assignment of the course, and that was reflecting on the discomfort that they saw within themselves throughout the entire course. So diving into these sub-themes here, first we saw coded language and institutional language. So this was seen in fear of failure, which we see a lot in healthcare students, feeling as though they have a lot to lose whenever they speak up. So fear of failure, so grabbing onto familiar topics, familiar words, such as equal opportunity to work hard towards personal aspirations, regardless of who you are and who you come from. So this might not have been an original thought. It was probably using a familiar thought process that they have been socialized to believe, and instead inserting that into what they feel about diversity instead of using their own original thoughts to answer that. And then we also noticed a silence of privilege and race. So students rarely, if ever, discuss these in their first time point of data. And it carried over into the responses by using like vague language to hint at privilege and race. So one of these examples was actually the title of my presentation, Distinctive and Unique People. Then we saw taking whiteness out of diversity, so disassociating themselves from diversity because most of the students in this class were white. And this was seen in a lot of responses, but I like this response that says, great, I have to write a paper on how I am diverse when I am one of the least diverse beings I can think of. So in time 2, this was the midterm assignment where they were to reflect on their initial responses and to also make goals for the rest of the course. So a lot of students noticed that they were uncomfortable with discussing these topics of cultural competency and diversity. And so they set goals to address discomfort. A lot of the reasons that they said they were uncomfortable was because they had never spoken about this before in a formal setting, or were afraid that their views would be unpopular among other students. So this was seen in the response here by student 2. I'm going to challenge myself to share what I'm thinking, even if I think it sounds stupid or it doesn't make sense. So, like I said, they also acknowledged and named discomfort, which is a huge step, as we'll go on to discuss here in a bit, in addressing that discomfort. So they explicitly named reasons of why they were uncomfortable in time 1. And part of this was because they came to a realization that they were participating in systems of oppression. So student 3 said, the area I'm most uncomfortable in is cultural humility. At the end of the semester, I want to say that this is where I am. I'm not done. This is something I struggle with, and I have to get out the checklist mindset. And then some students excuse themselves from continued discomfort by saying, yes, I was uncomfortable. I've now learned, and now I am culturally competent. I often find myself looking down on others because they are different, and that is a trait I want to eliminate due to how it makes me feel about myself, as well as it limits the possibilities that person and I could encounter. Okay, and so the last theme that we have is for time 3. So this was the final time point of data, and this was the final. So this was a final reflection, looking back at the course and looking back at the beginning response and seeing if they would want to improve anything about their diversity statement, saying how they contribute to diversity at the university. And so unfortunately, we saw some students who rejected the discomfort overall, saying that they think the I am diversity thing and how they contribute to diversity, as well as the course, was a hoax. Then we saw some students allowing discomfort. As I stated in the previous slide, that cultural competency is not a checklist item, and some students saw this as they've made improvements and now their work is done. So my goal is to not make assumptions throughout my future, especially as a healthcare provider one day, which we know is not necessarily feasible. And the goal here was to actually address these assumptions and to do better and know that this is a lifelong process. And so a few students did embrace this discomfort and embrace the idea of cultural competency as a lifelong process, as seen as in student 26's response there. So these tie into a lot of different theories. So one of these is white racial identity development. So this is a 6-stage developmental model aimed at understanding why white people maintain systemic racism and systems of oppression. As we can see, especially, I'm going to go back here, with student 27, their position throughout the entire course was not necessarily, I think this is a hoax. They actually went, you know, I'm going to buy into this for a little bit, and then went back to I am a hoax. So we can see that these stages are not always linear. And student 27 seemed to go back to the first stage of developmental model seen there. And then we also see the effects of transformational learning. So having these experiences, having a small group where they can debrief from the overall learning processes and the experiential learning and volunteering aspects of the course, that allowed them to talk with their peers, to talk in a non-judgmental format, and to go through why this is disrupting their cognitive frame. So why this is in direct conflict with what they've held previously, and to confront those assumptions. So implications. So this says a lot about active learning strategies and the importance of having that discussion-based group where students are not graded on their discussion, but having that to allow them to have those important discussions with their peers. the nature of conversations about diversity. As we saw in time one, some of these students, this was their first time even discussing how they contribute to diversity in a course. And then also the impact of institutions' diversity and inclusion mission statements. So again, in time one, we saw the adoption of these phrases used institutionally, and I believe it says a lot about the institution, how we address diversity and inclusion, because those are being absorbed by the students and how they conceptualize diversity. So some limitations, our research team, we were all women. So there was a lack of gender diversity in that. And also the class was one course over one semester. And it was made up of upper middle class white women and undergraduate students. Now this does represent the college as a whole, the demographics, but unfortunately that is not just, that limits us in our understandings. And then also the assignments were written data instead of verbal data, so we weren't able to better understand what individuals were discussing or talking about. We weren't able to follow up with questions, and we weren't able to see those nonverbal movements. So future directions. So health professionals defining diversity. We're in the beginning stages of conducting focus groups on health professionals who have taken this course and how that course has impacted them since taking it. And then having an interview component. Again, this is a focus group, so we're having verbal data instead of written data. And hopefully we're able to collect more information from participants who represent different genders and different social economic backgrounds. And here are my references. Any questions?
Amy, you do have a question in the queue.
Okay. Is it in the chat?
It is. If you cannot read it, let me know and I'll read it for you.
Yes. So diversity was not ever necessarily defined, I guess. It was more of a, oh, I should read the question. I'm sorry. So how is diversity operationally defined? More broadly in nature or primarily focused on race? So we actually asked the students what they believe diversity was. We gave them a prompt using an I Am Diversity video that the university had disseminated to students, which through that, it defined diversity as a broad variety of things, such as, race, ethnicity, gender, occupation, interests. And that is how our research team and the course aimed to describe it as, not necessarily just physical characteristics or something, or like race and ethnicity, but everything that makes you unique as an individual.
Great. Thank you, Amy. And in lieu of time here, unfortunately, I think I'm going to have to cut this session. Great job. Great presentation, Amy. Okay, and just as a reminder to the students, you have 10 minutes total, and that is a total of your presentation and Q&A. So if we can just be mindful of that so we can get everybody into this session, that would be fantastic. Thank you. All right. Our next presenter is Aly Peacock. Aly, are you ready to present?
I am.
Awesome.
I'll go ahead and share my. Okay, hi everyone. My name is Aly Peacock. My presentation is Comparison in Functional Measures and Adolescence with and Without Ankle Sprain History. This was done at the Sports Medicine Research Institute under the direction of Dr. Katie Bain. So lateral ankle sprains, or LAS, are the most common musculoskeletal injury. Additionally, most people sustain their first lateral ankle sprain during adolescence, but despite this, research regarding lateral ankle sprain has historically focused on the young adult population aged 18 to 35. Adolescents are actually an important population to consider because 60% of college-aged individuals report sustaining an ankle sprain at some point in their adolescent life. So in order to better understand the sequelae associated with LAS across the lifespan, more research is needed in the adolescent population because this is when the majority of initial ankle sprains occur. Young adults with a history of lateral ankle sprain exhibit decreased dorsiflexion range of motion, decreased reach distances on the STAR excursion balance test, and decreased health-related quality of life. And it is important to examine if similar deficits exist in adolescent population with a history of lateral ankle sprain. Therefore, the purpose of this study was to compare patient-reported outcomes and clinician-based outcomes between adolescents with and without a history of LAS. So in the bottom left corner of the poster, you can see the study design, the inclusion criteria, and the participant characteristics of this study. So the study design was a cross-sectional study, and the inclusion criteria were adolescents aged 15 to 19 years old, both with a history of lateral ankle sprain and with no history of lateral ankle sprain. In the middle column, you can see the patient and clinician-based outcome measures. And so for the patient-based outcome measures, participants filled out indexes that measured health-related quality of life. These indexes included the Foot and Ankle Disability Index, both activities of daily living and sport subscales, and the modified disablement of the physically active. The Foot and Ankle Disability Index is a region-specific measure of an individual's ability to perform simple tasks like walking upstairs and to assess joint stability of the ankle. The modified disablement in the physically active is a global scale used to assess the levels of disability in the physically active individuals. So for the clinician-based outcome measures, the weight-bearing lunge test and the STAR excursion balance test were used. The weight-bearing lunge test measures dorsiflexion range of motion, and the weight-bearing lunge, and the weight-bearing lunge test maximum distance reached between the foot and wall, with the knee making contact with the wall and the heel flat to the ground. So you measure the distance between the foot and the wall with the knee still having contact with the wall and the heel completely flat to the ground. So this was done 3 times, and the 3 trials were recorded and averaged. For the STAR excursion balance test, the static postural control of the participant was measured. Participants performed 3 reaches in the 3 directions shown in the images, and the average of these 3 reaches was taken and divided by the length of the standing leg. So for the statistical analysis, separate independent samples t-tests were formed to compare each primary outcome measure between groups. Significance was set at p less than 0.05. The results showed that there was no significant difference between adolescents with a history of lateral ankle sprain and uninjured controls in the clinician-based outcome measures, but there was a significant difference in the patient-based outcomes. The majority of research regarding health-related quality of life and physical impairment following lateral ankle sprain is performed on young adults, despite adolescence being the predominant time to experience an initial ankle sprain. It is important to learn about this age group. These findings demonstrate no significant difference between injury groups on the weight-bearing lunge test or the STAR excursion balance test. There was a significant difference between injury groups for the Foot and Ankle Disability Index, both activities of daily life and sport subscales, and the modified disablement in the physically active. So future research should aim to discover the effectiveness of implementing preventative measures for the adolescent populations to avoid initial lateral ankle sprain injury and re-injury. Furthermore, future studies should incorporate a longitudinal design to follow adolescents with a history of LAS across their lifespan. Again, it was found that adolescents with a history of lateral ankle sprain report worse region-specific and global health-related quality of life than uninjured controls, despite scoring similarly on the clinician-based outcome measures. That's it. Any questions?
Amy, you do have a question in the queue.
Okay. It's not showing up on mine. Maybe if I stop sharing.
Yeah.
Oh, wait, I found.
Would you like me to read it, or did you find it?
I'm not able to see it.
Okay. It's from Dr. Andretti. He said, were more for metrics, weight of subject, height, etc., similar across subjects?
There was a variety just because we mainly just focused on that adolescent population, so aged 15 to 19. But based on the fact that they were 15 to 19 and there was a broad range of them, they typically did fall in similar weight, subject, height, etc.
I have a question for you. Did you take into consideration how old the injuries were, and would that have any effect on the outcome?
We really just took into consideration how many ankle injuries they had had, not so much how old the injury was. At the point of taking their data, they had recovered from the lateral ankle sprain, so it would have at least been enough time for them to have recovered from the injury.
Got it. Thank you. Any other questions for Aly? Okay, great job, Aly. Thank you.
Thank
Do you
you.
plan to continue this research?
I do.
Nice. Okay. Well, we look forward to hearing more.
Thank you.
All right. Our next presenter is Maya. Maya, are you ready to present?
Yes, I am.
Okay, perfect.
All right. Hi, good afternoon, everyone. I appreciate you all taking the time to attend this presentation. Before I jump in, I just want to thank the Office of Undergraduate Research for hosting the showcase and my co-authors for their contribution and support throughout this process. My name is Mia Stefanelli, and the title of my project is Intra-Rater Reliability of Manually Segmenting T1 Row MR Sequences of the talar cartilage. To give you all some background, the talus is a bone in your ankle, and like many bones, it has articular cartilage protecting the surfaces, specifically where it interacts with bones, the other bones that make up the ankle joint. And this talar cartilage helps to protect from painful bone-on-bone contact in the ankle. However, following an acute ankle injury, such as an ankle sprain, the talar cartilage is prone to degenerative changes. And this outcome is called post-traumatic osteoarthritis, or PTOA. And the PTOA is a condition which affects over 5 million adults in the US and accounts for almost 12% of all lower extremity osteoarthritis cases. In fact, more than 3/4 of individuals with ankle osteoarthritis suffer from PTOA. Identifying early degenerative changes of PTOA can aid future investigations in developing new therapies to slow its progression. And essentially what happens in the progression of PTOA is a loss of key structural components in the cartilage, followed by an increase in water content and swelling capacity. And we can capture these early changes using compositional magnetic resonance imaging, or MR, sequences, such as T2 or T1 row, the latter of which was used in this study. However, currently the only way to quantify these MR sequences is through manual segmentation, and limited information is available on the reliability and reproducibility of that process. So the purpose of this study was to determine the intra-rater reliability of manually segmenting, T1 row MR sequences of the talar cartilage. The study was of case series design, and our subject demographics can be found down here in table 1. This information includes sex, age, height, weight, BMI, and the ankle imaged. Manual segmentations of the T1 row MR sequences of the talar cartilage were performed using ITK SNAP software with the initial and repeat segmentation times separated by 1 week. An anatomical reference point was used to mark what was determined to be the talar cartilage, voxel by voxel, for each slide of the T1 row sequence. And that adds up to the total volume. If you look here at figure 1, you can see one slide of the T1 row sequence in ITK SNAP with the individually selected voxels shown in green and yellow. The slide from subject 1 is compared between the initial and repeat segmentation times. The segmented cartilage for all times and subjects was then divided equally into 4 regions of interest, or ROIs. Anteromedial, which was labeled red, anterolateral green, posteromedial blue, and posterolateral yellow. These ROIs can be seen here in figure 2, which displays 3-dimensional renderings of the talar cartilage produced automatically by the software system. And these images are compared for all 3 subjects between the initial and repeat segmentation times. For our results, the 2 measures of interest, talar cartilage volume and mean T1 row relaxation times, are displayed here in table 2 for each of the 4 ROIs and for both segmentation times. The strength of the reliability of these data is shown through interclass correlation coefficients, or ICCs, and the significance through their respective p-values. Our data demonstrated good to excellent intra-rater reliability for manually segmenting talar cartilage volumes and calculating mean T1 row relaxation times for all ROIs, as well as significant p-values for all ROIs with the exception of anterolateral. This shows that while some improvements can still be made, manually segmenting T1 row MR sequences of the talar cartilage is reliable. Demonstrating the reliability is important for future prospective studies investigating changes in talar cartilage composition following an ankle joint injury. And this is primarily because quantifying those early compositional changes in that structure of the talar cartilage can help identify patients with or at risk for developing PTOA. Looking ahead, future research will need to additionally determine the inter-rater reliability of manually segmenting the talar cartilage volume and calculating mean T1 row relaxation times. But to conclude, manually segmenting the talar cartilage volume and calculating mean T1 row relaxation times is reliable and reproducible. Thank you so much for your attention. I'll be available for questions if you have any, but you can also go to my symposium page and leave questions or comments there.
You do have a question in the box. Can you see it?
Hold on. Is it in the chat?
Yes, I believe it is.
Okay. Let's see. I'm not sure. Okay, the question was, can this method be used to characterize different forms or etiologies of OA? I don't know yet at this time, but if you're still curious, I can get back to you afterwards.
So I have a question for you, Mia. Do you plan to continue this work?
Yes, I do.
So you are, what year are you?
I'm a junior.
A junior. Okay, nice. I had multiple ankle sprains as a high school athlete. And so a little bit of that I've seen the effect of this slightly. Mine is not real bad. So this is very interesting to me personally.
Yeah, I think everyone, well, a lot of people can relate to this because ankle sprains are so common.
Mm-hmm. Mm-hmm. It would be interesting to see if how you can, especially in adolescence, how you can help refrain from getting so many, because I had multiple ones, reoccurring ones as an athlete. So I'm sure they've come a long way since I was an athlete, but be interesting to see. You do have another question here. Can you see that one?
Oh, this in the Q&A?
Oh, actually, it was just a comment.
Okay.
Just said, great work. I know cleaning the data is tedious work. Did you find that tedious?
Sort of, especially at first, because it is just, individually clicking boxes. but it's really cool to see it all come together. Like, I think that the first time I saw like the image, like the 3D image, like of it all together, it was just kind of like mind blowing, like that all of the boxes came together for that.
Does anyone else have any other questions for Mia? Well, great presentation, Mia, and good luck in your future research on this.
Thank you.
Okay. All right, our next presenter is Jenna Wilson. Jenna works with Dr. Matt Hodge in health sciences, athletic training. Jenna, are you ready to present?
Yes, I am.
All right. You're welcome to share screen.
Okay. Okay, so can you see this? Hi, my name is Jenna Wilson, and for this study, I looked at the executive function in Division I collegiate athletes and recreationally active college students. So sports require the use of high-level cognitive functions such as attention, decision-making, and memory, all of which are part of the executive function domain. Currently, it's unclear whether or not high-level athletes exhibit similar executive function compared to recreationally active athletes. So therefore, the purpose of this study was to compare the executive function of male and female Division I high-level athletes with that of their recreationally active peers. Neurocognitive tests are an important tool used to compare cognitive function across individuals of the same age or to compare cognitive performance of the same person at different time points. The National Institutes of Health Toolbox, commonly referred to as the NIH Toolbox, is a computerized neurocognition test that can be used to assess processing speed, cognitive flexibility, attention and inhibitory control, and episodic memory. Specifically for this study, 2 tests from the NIH Toolbox cognitive battery were used in order to test cognitive flexibility and attention and inhibitory control. The subjects for this study were 37 Division I athletes and 30 recreationally active college students. All of the NCAA athletes in this study were actively participating in their sport and were cleared for full participation at the time of the study. And participants were excluded from the study if they were currently recovering from a musculoskeletal injury or concussion, experiencing post-concussion symptoms, they had a history of neurological or vestibular disorders, or they experienced motion sickness or vertigo. Participants completed the flanker inhibitory control and attention test and the dimensional card sort test from the NIH Toolbox cognitive battery on an iPad. In the middle column of the poster, you can see the flanker test assesses visual, attention, and inhibitory control. This test presents participants with a series of arrows in congruent and non-congruent patterns and asks them to identify the direction of the middle arrow as quickly as possible. The dimensional card sort test assesses cognitive flexibility by presenting the participant with a stimuli and asking them to match it with a similar shape or color as quickly as possible. Fully corrected standard scores were calculated for each subject to correct for state, sex, age, education, race, and ethnicity. Two-way ANOVAs compared cognitive performance based on group and sex. In the results of this study, we found that there is a significant group by sex interaction on the flanker inhibitory and control attention test. Division I female athletes scored lower on the flanker test than Division I male athletes. There was also a significant sex effect, with Division I females scoring lower again compared to recreationally active females. There was not a significant difference on the flanker test between Division I males and recreationally active males. And similarly, there was no difference between group by sex, or group on the card sort dimensional analysis test. In conclusion, female Division I athletes performing lower on the flanker test than male Division I athletes and recreationally active female athletes may indicate a decreased ability to suppress motor responses caused by internal or external distractions during sport. These findings are important for identifying possible avenues of intervention in hopes of mitigating injury risk
as female athletes are more prone to many sports-related injuries than our male athletes. The significance of these findings for improving athletic performance should also be explored further.
All right, great job. Any questions for Jenna? So Jenna, do you plan to continue this research?
I actually just started a new project, the same lab, same type of study, but it's a different project other than what this was looking at.
Sure. Okay, you do have a question. Can you see it? And if not, I'll read it to you.
Oh, yes, I do see it. So yes, reaction times were recorded and compared for that. So the average of those were taken. We didn't really look at specific times like themselves. They were recorded and we did like look at that data, but we really just ended up taking the means of all of the scores that were given to us.
Any other questions?
Oh, it looks like there's one more in the other chat. So the question was, what do you feel this relationship would look like in those with history of concussion? I've talked about this a little bit with my mentor. I think that if in history of concussion, you would still see the same trends that are seen here in the results, but I think that the trends of the difference between groups would be smaller. So I still think that Division I females would score lower on all the tests. The difference between Division I females and Division I males might be a little bit smaller, and the same thing for all the other groups. So I still think the same trends would exist, but just the difference between the groups would be smaller than those trends. There's another question that said, why do you think that there were differences between males and females, especially for female Division I athletes? That's a great question. And that kind of, we were looking to see why that might be. So females do have they're more prone to sports injuries. So I think that might be a reason why this difference was there, is just they have a little bit more distraction for either internal or external distractions during even these tests, especially sport, which leads to those injuries. So this was kind of the results that we expected to see when this study. This is what we expected we would find. So I think that just overall is just the trend of athletes in general, and even the recreational active athletes, that was still seen in them. So I think females just have a lot more, they don't suppress their distractions as well as males do in sport related activities.
You do have another one, Jenna?
Working memory. Do you think working memory plays a role? Yeah, definitely. So I think you would see too that I think if your working memory was greater, you wouldn't necessarily have to process things as much, because you would kind of have a little bit more like just natural working memory to use off of. But I still think that even if you saw that working memory was higher in females than it was males, I still think that this trend would stay consistent.
Awesome job.
Thank you.
Okay, we will move on to our last presenter. Next will be Noah. Noah, are you ready to participate
I am ready.
and present?
I am
Okay.
ready.
All right, okay, you can share your screen.
Awesomely. Find which.
Yep.
Can everyone see it well?
Yes.
Make sure nothing's cut off for you guys. Awesome. Well, first, thank you guys for putting this on. I know it's very difficult, especially with COVID, trying to get everything coordinated and Zoom links sent and all that. So I appreciate that from you guys. And I just want to thank Dr. D just for all the work she's done with me over the past year, especially with COVID, staying involved with me and continuing to help me get more and more involved in research and learn more about it. Just means a ton, especially with me graduating this year. I just want to say that thank you to her. So ultimately, on behalf of my co-authors, I'm pleased to present our research on injury history, sports participation, and current physical activity among young adults. So we know that youth sports participation is associated with short-term injuries, and we also know a little bit about the long-term relationship, but there seems to be a little bit of a gap in the research between those that get injured in high school and how that plays 4 or 5 years down the road, specifically in the ages of 18 to 25. So the purpose of this study was to examine the association between injury history and current moderate to vigorous physical activity among young adults. So to collect our data, we use research match and university classes to help gather participants who completed REDCap surveys, where they answered things related to their demographics, injury history, what injuries did they have, self-reported severity scores, how long were they out for, as well as what sports did they play in high school, and were they competitive high school athletes, recreational athletes, or non-athletes in general. So to be eligible for this survey, the participants had to be 18 to 25 years old, not currently injured, and have no physical activity limitations at the time. To measure it, we used the survey and we calculated using the Good and Leisure Time Exercise Questionnaire, which is a validated study, which we calculated MVPA, or moderate to vigorous physical activity, by the formula shown here, ultimately. And that allowed us to create a two-way ANOVA between moderate to vigorous physical activity and high school status, as well as high school and previous injuries. So ultimately, our demographic was roughly around the age of 22.2 years, primarily female and white. And we were able to, in the overall, have a pretty even 0, 1 to 2 or more injuries overall in injury status. So we were able to create this graph, which helps to show our data, with moderate to vigorous physical activity on the left. And to also be considered physically active, they had to score a 24 or above on the Good and Leisure Time questionnaire score. We broke them up based on injuries. So we had those with 0 injuries, 1 injury, and 2 or more injuries. And as well, we broke them up based on if they were a high school athlete or a non-high school athlete. And we also grouped in our non-high school athletes, we grouped in recreational athletes, specifically because when we looked at the data overall, comparing them, they fit their curve appearance fit most with the non-high school athletes than they did with the high school athletes. Through that, we were able to gather results that as the frequency of injury increases, as the frequency of injury, as there are more frequent injuries for non-high school athletes, higher levels of moderate to vigorous physical activity were found. And then we saw the reverse relationship with high school athletes. So as the frequency of injuries increase, lower levels of moderate to vigorous physical activity were found. And we found for 2 or more injuries, participants had similar levels of moderate to vigorous physical activity. And it did not seem to matter if they were a high school athlete or a non-high school athlete. We were able to find a statistically significant interaction between injury frequency and high school athlete status on current moderate to vigorous physical activity. And as I said before, among individuals who reported 2 or more injuries, similar moderate to vigorous physical activity for former athletes and non-athletes were found. Through this, we ultimately were able to conclude, we were able to conclude that, sorry, let me rephrase that. So ultimately at this point, we're working on looking more into the explanation of why we see these curves like they are, whether that be, is it because of severity or other external factors which we have not yet looked into. And ultimately, in conclusion, efforts to reduce risk of injury during youth sports participation may be associated with long-term physical activity participation. And as I said before, future studies should examine whether young adults who have experienced multiple injuries have unique physical activity barriers or facilitators, and also to identify if injury severity is a contributing factor for less moderate to vigorous physical activity. And I also just want to say a thank you to also my other co-authors, Chloe McKay, Johanna Hodge, as well as Jennifer Tinsley, who has just recently joined on.
Nicely done, Noah. So you do have a question in the chat.
Okay, I
Let me-
cannot see it. So
Okay, I will read it to you. Sure.
Thank you.
It's from Dr. Andretti. He said, did you notice differences in the high school athletes depending on the specific sport they were involved in?
We have not looked into that yet. Our current plan right now is what I'm in the process of helping to lead author this paper for publishing. So we have not looked into that statistic yet. We did notice that specifically for our 2 or more injuries, we had less, ultimately less participants for 2 or more injuries compared to the other injuries. But we have not yet looked into if a specific sport is like more, if they say they played football, they are more likely to have injuries and more likely to be more moderate to vigorously physically active. We just have overall looked at the general, just overall in general, but we have not looked into the specifics of that yet.
I think that's something that could be investigated in the future and kind of broken down.
Yes, we're in the process of currently turning this into a paper, and we're in the process of going through and reanalyzing our data to make sure and look at more things. So that's definitely something we will look into and see if it does play a role.
So Noah, you mentioned it. Did you mention this is your last year?
This is my last year, sadly. So I'm graduating this May, and plan is to continue to work on this research until it gets to publishing, which we're hoping to by the end of May.
Nice. Very nice. Any other questions for Noah? I think you do have a question here. I think this was yours. It says, why do you think there were differences between males and females, especially for female D1 athletes?
I believe that was for the presentation.
That was the okay.
Yeah.
That was the previous presentation. Sorry about that. They don't go away, so I have to, my bad. I did not pay attention.
You're fine. Don't worry about it. I've noticed that too. Looking in, I'm like, okay, which presentation does this line up with best?
Mhm. All right. Well, if there are no more questions for Noah, that concludes our presentations for this session. I would like to say to all the student presenters, thank you very much for presenting. For those of you graduating and moving on, good luck to you, Godspeed to you. And I hope you have future success, whether you continue your research or not.
Awesome. Thank you guys very much.
Thank you. All right, this concludes our presentation.